Hi guys, today I'm going to present my journal that title of Effects of High Doses of X-ray on Hematological Parameters and Morphology of Red blood cells in Human Blood. The authors are Carmen Faraj, University of Soleimani, Ali H. Taki, Sarah A. Zainar from University of Kuruk is College of Science. I'm the presenter Subash Raj 40030037141D is my matrix number. Next slide. Introduction. The effects of ionizing radiation on people can arise from both low and high doses. The interaction of radiation with the DNA of the cells may cause the radiolysis of water inside the cell producing free radicals. The penetration of radiation into living cells can result in the transfer of radiation energy to the biological material. The absorbed energy can induce the formation of reactive oxygen species, chemical bond breaking, and ionization of different biological essential macromolecules, for example, DNA, membrane lipids, and proteins. The DNA damage induced by radiation, such as base alteration, cross linking, strand break, chromosomal aberration, can lead to mutation. Radiation doses to radiosensitive organs, such as the ovaries, testicles, and thyroid, may induce harmful effects. For example, cancer and genetic effects. My objective of this journal is the objective of this journal is to investigate the effects of X-ray radiation on some hematological parameters, that is morphology of red blood cell RBC, erythrocyte sedimentation rate ESR, analysis of human blood using computed tomography CT scanner, the literature review. Electromagnetic radiation that originates outside of a nucleus is known as X-ray. It has less energy than gamma ray. The majority of X-rays are created artificially, and they are mostly used in medicine. Atoms can get ionized by ionizing radiation when one electron is removed from the orbits, and it creates ions with a positive charge and free electrons with a negative charge. Ionizing radiation encompasses radiation produced by both naturally occurring and artificially created radioactive elements. Ionizing radiation can cause cellular damage at both low and high doses depending on the target, dose, rate, sex, age, and kind of target. Direct interaction refers to a method where ionizing radiation interacts directly with important targets such as DNA. It is also known as indirect contact when ionized radiation interacts with water in a cell to create both ions and free radicals. It's by Taki Ali Faraj Zainal Zara 2018. The primary effect of radiation on living tissues is the modification of cellular chemical characteristics. This modification may be related to separation of amylotic molecules and the formation of secondary electrons, which results in the generation of free radical roots and hydrogen peroxide in bodily fluids. These effects can directly rise as the radiation exposure increases, although radiation can harm living cells. When used correctly, it can be beneficial to our lives. Even at modest dosages, X-rays have a great sensitivity in the blood. Radiation impacts could be influenced by a variety of factors, including exposure of time and live organism age. Several hematological alterations were observed with chronic radiation exposure. Moving to the results, there are four tables that indicate the results. Okay. Uh, Tables 1, 2, 3, and 4 illustrate the results related to the measurements of the hematologic parameters in the control group, and 4 experimental groups irradiated with 3, 6, 9, and 12 GY. There was no significant difference between the control and experimental in terms of all parameters. As you can see, WBC, neutrophil, lymphocyte, phonocyte, eosinophil, basophil, RBC, HP, HCT, MCV, MCH, MCHC, RDW, and platelet. Okay, moving on to the effects of morphology. Um, After irradiation of blood to 369 and 12 GY X-ray, the percentage of RBC crenation were less than 5%, as you can see clearly, and 50% and 40% and 60% respectively. Figure 1, 2, and 3 and 4 represent the crenation study of the control and 4 experimental groups. The results, the crenation analysis revealed an explicit morphological changes. Okay, discussion. The effects of exposure to ionizing radiation on individuals' health status were vary depending on the amount of absorbed dose and type of cell. In this study, there's no significant changes were observed between the control and 4 experimental groups in terms of hematological parameters. It was found that some characters of RBCs are such as a concave shape by a phospholipid bilayer cytoskeleton were related to the some erythrocyte disease variation in the packing of properties of these molecules may result in changes of intramolecular van der Waals forces forming the cellular membrane, which in turn alters the membrane malleability and morphology. These increases of crenation of RBC with increasing doses and the decreases of radius indicate changes in morphology after irradiation. Scores et al. Investigate the irradiation of RBC to 450 GY X-ray cause aggregation of membrane proteins. Ferrell et al. Examined how radiation exposure can damage living cells and modify others. Shishikina et al. Concluded that exposure to low doses of gamma and X-rays show a high sensitivity of the characters of lipid metabolism in erythrocytes. Hal-Ainim et al. Studied erythrocyte of rats to investigate the effects of X-rays on RBC membrane solubilization by using sodium diadesyl sulfate (SDS). In vivo investigation, blood film showed regularly shaped red blood cells, while those photographed 10 days post irradiation revealed some sort of repair. Moving on to conclusion, ionizing exposure can result in stem injuries of hematopoietic system, one of the radio-sensitive systems in the human body. This type of damage may lead to alteration in the production of bone marrow, stromal cells. Moreover, it known as that exposure to ionizing radiation has direct detrimental effects on blood cells. Blood cells respond to the radiation in a variety of ways, which may result in different amounts of the effects. It was observed that in the vitro irradiation of human blood cell to different X-rays doses, for example 369 and 12 GY, like in our journal, resulted in the enhancement of RBC crenation with increasing the dose and reduction of the cell radius compared to the dose in the control group. However, the hematological parameters and ECR analysis were not statistically affected. My personal comment about my journal is the blood is highly sensitive to X-ray even in low doses. Different factors could play an important role in radiation effects such as the exposure time and living organism age. It was seen that there are several hematological changes during chronic exposure to radiation. Thank you for listening to my presentation. Have a great day.